So this week we're going to talk all about how we can kill pathogens and bacteria and fungus. So the first chapter we're going to discuss is chapter 11, and this is basically how we can use disinfectants and chemicals to kill microbes on surfaces and how we can use it to prevent infections. And then in chapter 12 we'll talk about antibiotics and how we can treat already established infections. So first, let's talk about how easy or hard is it to kill different types of microorganisms. By far, the hardest organisms or hardest forms of microbes to kill are the prions. Remember, prions are those infectious proteins, and they're extraordinarily difficult to destroy. And then followed by endospores. Endospores are those dormant, heat-resistant forms of bacillus or clostridium, and they're found, everywhere on all surfaces. So we really have to be careful with them. So these two are, especially the endospores, because they're everywhere, difficult to destroy. Now, the easiest thing to kill, the least resistant forms of microbes, by far the easiest pathogens to kill are any type of gram-positive bacteria that are not endospores, so the vegetative cells. Remember, vegetative cells indicates they're actively growing. So actively growing gram-positive bacteria are pretty easy to kill. In addition, fungi, active fungi, the fungal spores, yeast, enveloped viruses are pretty easy to destroy, and protozoa and trophozoites, the actively growing forms. Kind of in the middle now, the gram-negative bacteria are a little bit trickier to get rid of because remember they have that second outer membrane. Pseudomonas species in particular, these are gram-negative. They're highly resistant. Mycobacterium tuberculosis, the acid-fast bacteria, pretty tricky to destroy. Now, Staph aureus, even though it is gram-positive, it's notoriously resistant, especially against antibiotics. And finally, those protozoan cysts are kind of in the middle, those dormant forms of the protozoa. So what the main takeaway from this slide, endospores, extremely hard to kill. Gram-positive and vegetative, actively growing bacteria, easier to destroy. When a bacteria is actively growing, it's more easy to destroy than if you have like a dormant or an older unactively growing cell. This table is showing you the only ways that we can actually destroy endospores. The most important and most common way we destroy endospores is using the autoclave. The autoclave is a moist heat method. Moist heat is very effective. So we will autoclave at 120 degrees Celsius for usually about 30 minutes, and it's also about 21 pounds of pressure. This is enough to destroy endospores. Now, some equipment, very heat-sensitive equipment or medications, vitamins, antibiotics, we obviously can't autoclave these things because the heat will destroy them. So some alternative things for sterilizing, we can use X-rays. This is a form of radiation. We can use this one type of gas that's a sterilant. It's called ethylene oxide. It's extraordinarily dangerous, so usually if a hospital will have an ethylene oxide chamber in a specific location where they can sterilize this sensitive equipment. Or there's one sporicidal liquid chemical. You could use glutaraldehyde, again, an extraordinarily toxic chemical. We are very, very careful when we handle it because not only will it destroy endospores, it can also destroy vegetative cells, including human cells. So these methods are the only methods we can use to sterilize. When you see sterilize, that means we've ensured destruction of all life, including endospores. Now here are some important terms, all right, and I'm going to kind of go through the main differences between them all. Like we know, sterilizing indicates destruction of all life, including endospores. Now, whenever you see a term that says cidal, such as microbial or germicidal, cidal indicates to kill. So this is a method that we can use to kill the microbe, whether it be a bactericidal agent, fungicidal, sporicidal. Now, microbial agents indicate we're killing vegetative microbes and not the endospores. Disinfection indicates that we are eliminating most vegetative cells or pathogens from usually an inanimate object. So if it's a disinfectant, We usually reserve that for something that's going to go on a surface or, a piece of equipment, something like that, a physical or chemical agent used on an inanimate surface. Remember, it's going to destroy mostly vegetative pathogens, but not those endospores. Similarly, an antiseptic is similar to disinfectant, but it's safe for human tissue. So something you can put on skin or over before surgery or any type of bodily surface, and sometimes like a mouth antiseptic, like an oral antiseptic. This will destroy or inhibit the growth of vegetative pathogens on human tissue, but again, it does not kill endospores. Some agents are microbiostatic. So whenever you see static, it indicates to basically stop the growth or temporarily prevent the growth of the microbe. So for example, when you freeze your food, freezing is microbiostatic. You're temporarily preventing growth of microbes on your food, but you're not killing them necessarily. There's also some methods where we may reduce the number of microorganisms. It doesn't necessarily kill all the pathogens, it just reduces them. These are more kind of mechanical methods, like you're actually scrubbing away debris. So for example, sanitation is any technique where you're mechanically removing debris from an inanimate surface. So we usually reserve sanitation for non-living inanimate surfaces. For example, when you wash your dishes or you do your laundry, you're physically removing debris from those surfaces. Whereas degermation is same thing, but you're removing debris from human tissue. Like when you wash or scrub your hands, you're mechanically scrubbing away debris on your skin. All of these terms are condensed in this nice chart. This is a good chart to have, print out and study hard. There's a lot of factors to think about when you're deciding how to treat an object, whether it's safe to sterilize, okay? Can it be heated in an autoclave? Is it sensitive to certain chemicals? Does it need to be sterile or can we disinfect it? We also got to think, how many microbes are present on this? What kind of microbes? Do we expect a lot of endospores? Is this something that's going to penetrate into the inside of a human tissue? Or is it just going to be surface associated on skin? We might not necessarily need it to be 100% sterile. So there's a lot of factors to consider. In terms of antimicrobial action of various chemical, gaseous, or physical methods of destroying bacteria, there's basically 4 targets. And when I say targets, remember, we need some way to kill the microbe or prevent it from growing. We need something that'll target the microbe and eliminate its growth. So there's 4 main targets. The first main target, and the one we'll probably talk about a lot, especially in the antibiotics chapter, is the cell wall. Remember, this includes the peptidoglycan, any proteins in the wall, synthesis of the cell wall, the actual structure of the peptidoglycan or the LPS itself. And this is really important because the cell wall is on the outside of the bacterial cell. So it's the easiest to target, and without the cell wall, the bacteria can no longer be intact. So a lot of the drugs we'll talk about target the cell wall. Detergents, alcohol, these are just some examples of agents that will destroy the cell wall. Next, the cell membrane, because of course the cell membrane is also on the outside of the cell, so it's an easy target. And if we destroy the cell membrane, then again, the cell can't function. Now, any agent that destroys the integrity of the cell membrane typically fits in this category. We call it a surfactant. So if surfactants destroy the cell membrane, here's how it works. Here's the cell membrane. You can see the phospholipid bilayer. These little green things are the surfactant molecules. The surfactants basically insert themselves into the membrane, destroying its structure. Now you can see the membrane has all these openings that can allow molecules in and out of the cell, and the cell will eventually no longer be able to survive. The final two main targets are the protein and nucleic acid synthesis. So this includes any enzyme, ribosomes, anywhere that's involved in making DNA or RNA or making proteins in the cell. So again, a lot of drugs like, for example, chloramphenicol, which prevents protein synthesis. UV light actually destroys DNA of the cell. And formaldehyde, a chemical, you guys probably have seen this before because it's used as a preservative. And the reason they use it is because it destroys bacteria. And then the final target is actually destroying proteins themselves, destroying fully formed proteins. A lot of chemicals act to destroy proteins. For example, alcohols, phenols. You find phenol in, for example, Lysol, acids, and heat. There's three main ways that these agents can destroy the protein. The first, for example, heat or acid, does what's called denature the protein. So here's the protein in its normal shape. When it's denatured, it basically becomes unfolded and basically melts and it can't work properly. Similarly, denatured proteins could take on a different shapes or melted so that they can't work anymore. Or some drugs, or for example, metals, heavy metals, they actually can insert themselves onto the protein, blocking the active sites, especially of enzymes, and now the proteins can no longer function properly. There's three main methods we use to control microbial growth. First, we could use physical agents. Physical agents include heat, and we separate this into dry heat and moist heat being much more effective than dry heat. And then the second main physical agent is radiation. And radiation could include X-rays, which are ionizing radiation, or UV light, non-ionizing. The second main method is a mechanical removal, which basically means filtration. In filtration, we're not actually killing the microbe, we're just removing it from whatever our sample is. And then finally, chemical agents. Chemicals could either be in a gas form, such as the ethylene oxide we talked about, or a liquid form like common disinfectants we use in everyday life.